Assalamualaikum and hello everyone. Today, I will present about theory of genetics and inheritance. This topic focuses on how traits are passed down from parents to offspring through gene. This story begins with a scientist named Gregor Mendel. He was an Austrian monk born in 1822 and is known as the father of genetics. Mendel did experiments with pea plants between 1856 and 1865 to study how traits like flower color and seed shape are inherited. Even though his work was ignored at first, it became the foundation of modern genetics after being rediscovered in 1900s. Next, Mendel discovered three important laws. First, law of dominance. Some traits are dominant, others are recessive. Second, law of segregation. Each organism has two alleles, but only one is passed to each offspring. Lastly, law of independent assortment. Genes for different traits are passed separately. These discoveries help us understand how traits are inherited and why we look different from each other. Before we continue, let's understand some key terms. First, gene, a segment of DNA that controls a trait. Second, allele, different version of a gene. Genotype, the genetic makeup of an organism. Phenotype is the physical appearance like tall or short. Heterozygous means two different alleles, while homozygous means both alleles are the same. Now, I want to explain the law of dominance, which is one of Mendel's important discoveries. Let's say we have two pea plants, one tall and one short. When we cross them, all the baby plants in the first generation, or F1, will be tall. This is because tall is a dominant trait. Even though the short trait is there, it is hidden. So, all F1 plants look tall. But when we cross the F1 plants with each other, the second generation, called F2, will have both tall and short plants. This shows that the short trait was still there in the F1 plants, just not visible. So, a dominant trait only needs one copy to be seen, but a recessive trait needs two copies to appear.
Hi, I'm Hana. I'm going to start with Mendel's first law, also formerly known as the law of segregation. This law explains how traits are passed from parents to offspring. This law is built upon three core principles. Firstly, every individual's diploid organism, like us, possesses two alleles for each specific characteristic. When gametes are formed, these two alleles segregate or separate so that only one allele ends up in each gamete. Secondly, this separation of alleles during gametes formation is completely random. Each gamete receives just one allele for a given characteristic, to ensuring the genetic variation. And thirdly, during fertilization, the offspring inherits one allele from each parent. This recombination restores the diploid state and leads to the unique combination of traits in the new individuals. This random segregation is crucial for the genetic diversity, so we can see it in population. Let's explore the monohybrid cross, which is a practical application of Mendel's first law. A monohybrid cross is a genetic cross specifically designed between two true breeding parents that differs in only one characteristic. The term true breeding is vital here, meaning that if these organisms were to self-fertilize, they would consistently produce offspring identical to themselves for that specific trait. To F2 gametes, when F1 individuals are crossed with each other, we can predict the following distribution of genotype in their offspring. 1 out of every 4 offspring will be homozygous dominant, and 2 out of every 4 will be heterozygous, while 1 of every 4 will be homozygous recessive. Now that we have covered the monohybrid cross, let's discuss how we quantify the result of this cross by differentiating between genotype and phenotype ratios. These ratios are crucial for predicting the genetic makeup and observable traits of offspring. The genetic ratio describes the proportion of different allele combination or genotypes that present in the offspring. It tells us about their actual genetic blueprint. The phenotype ratio, on the other hand, describes the proportion of different observable traits in the offspring. This is what we can physically see or measure that the outward expression of the genes. Next, let's explore how probability works in genetics. There are two main rules we use here, the addition rules and the multiplication rule. First, the addition rule. This is used when we want the probability of either one event or another occurring. Then, we have the multiplication rule, which we use when we want to know the probability of both events happening together. Finally, let's explore a very practical and important technique in genetics, the Testcross. A Testcross is an experimental cross used to determine the unknown phenotype of an individual that display a dominant phenotype. We know it has at least one dominant allele, but we don't know if it is homozygous dominant or heterozygous. Thus, the strategy of a Testcross is to cross the unknown dominant individuals with a non-homozygous recessive individuals, and the offspring phenotypes will then reveal the unknown parent's genotype.
Lastly, I will explain on our last Mendel's law, which is law of independent assortment. The law of independent assortment states the genes for different traits are inherited independently of each other during gamete formation. This involves dihybrid cause will focus on the transfer of a single characteristic. The law increases the genetic variation in the offspring. In F1 generation, all offspring shows the dominant traits for both genes, while for F2 generation, it produces offspring with various combination in phenotypic ratio with 9 to 3, 3 to 3, and 3 to 1. This is a basic explanation of the law. Now, let's look in more details at the dihybrid cross. The alleles of the gene set AA will assort independently of the two gene sets. It's not mandatory that all the dominant alleles will get assorted in the cells together. So, the allele capital A can get assorted with any allele from the gene set BB and CC. That's the reason why we get the gametes with different possible combination at the end. To quickly review, the assessment of alleles is purely random and does not have a predictable and fixed pattern during the process of gamete formation. That is what the law says. There can be more possibilities of obtaining different combinations depending upon how the chromosomes align themselves on the metaphase plate. A dihybrid cross illustrates the potential results from crossing individuals that differ in regard to two traits. In this case, the parents or P generation differ in both P color and shape. A plant homozygous for wrinkled green peas is crossed with the plant homozygous for round yellow peas. Because both parents are homozygous for both traits, only gamete coating for wrinkled green peas or gamete coating for round yellow peas are produced. A diagram of the cross between two members of the F1 generation, called a Punnett square, is shown here. When the gametes from these pea plants are combined, all of the offspring in the F1 generation are round and yellow. This is because the traits of for round and yellow are dominant over the traits for green and wrinkle. Because all offspring within the F1 generation are heterozygous for both traits, both the males and females can produce four different gamete combinations: round yellow peas, wrinkled yellow peas, round green peas, and wrinkled green peas. A Punnett square is created to determine the F2 generation that result from this cross. Of the offspring produced within the F2 generation, 9 are round yellow peas, 3 are round green peas, 3 are wrinkled yellow peas, and 1 is a wrinkled green pea. In other words, a 9 to 3, 3 to 3, 3 to 1 ratio exists within the F2 generation. Mendel was able to use this observation to determine the factors assert independently of other pairs of factors. This has become known as the law of independent assortment.